Hello, everyone. Welcome to the third installment of our High Elevation 5 Needle White Pines 2024-2025 Science and Management webinar series. I'm Enzo Martelli, a PhD student in Karen Nelson's lab at the University of Montana, and I'm co-organizing this webinar with Lou DeLucy, a PhD student in Daniel Ulrich's lab at Montana State University, and Karina Lee, a master's student in Diana Tambak's Forest Ecology lab at CU Denver. We have another great presentation for today, but before that, I'd like to start by introducing our chair, Glenda Scott, who will share a few announcements with us. Welcome, Glenda.
Hi. Hi, thanks you guys for having me. I was on last week, so you might have heard my introduction, but we have a emphasis in the Whitebark Pine Ecosystem Foundation for all high elevation five needle pines, and also to support student research. And so one of those opportunities, if you're a master's or a PhD student conducting research and writing a thesis on any high five pine, we hope that you will look at this grant proposal, grant opportunity, I should say. I'll say it again at the end too, but the RFP is due February 14th, so you have a couple of months. And in the message box will be the link to the grant application, which is on the Whitebark Pine Ecosystem Foundation website. So if you've perhaps in the academia world, you've met many of the folks that, including Enzo and Lou and folks that have received the grant. It's $1,000 to $2,000, best used for your research equipment and that kind of stuff. It's both Canadian and US, a joint grant program. So Whitebark Pine Ecosystem Foundation has a Canadian sister organization, and we manage this grant together. If you've been to our annual meetings, what funds it is the auction. So we have funded some students from British Columbia, and so it can be both Canadian and US academic programs. We will ask that you present your findings at our annual meeting, and hopefully present at our Nutcracker note publish in our Nutcracker Notes. It's a biannual digest of research and other information. So we want you to share it. We want to support student research and all of you. So we hope that you'll look at that opportunity. And this having Kristen today is a great opportunity or exposure to other high five pines in addition to whitebark. So with that, I hope that you'll check out our website. Again, February 14th is the due date for your application. So thank you
Thank-
very much for attending, and this is such a great seminar. It's nice to be part of it.
Thanks so much for joining us today, Glenda. So if you miss any of our previous installments, you can find recordings of the talks on our YouTube channel, and the link to our website will be posted in the chat shortly. And finally, after the presentation, we will have a QA session where you can share your comments, doubts, and questions. We will not have a QA, we will not be using the QA tool in Zoom. Instead, please post your comments on the chat box and our team will be collecting them. And for those comments, we really recommend you to give context on the slide or part of the presentation you're referring to, just to make it easier for our presenter and also the rest of our team. Now, it is my pleasure to introduce today's speaker, Dr. Kristen Waring. Dr. Waring is a professor of silviculture and applied forest health in the School of Forestry at Northern Arizona University. She specializes in finding solutions to challenging and complex forest health problems by incorporating interdisciplinary research into silvicultural solutions and other management strategies. Dr. Waring received her BS and MS from University of Montana in Forestry, specializing in applied forest entomology for her master's research project. She then earned her PhD from the University of California, Berkeley in silviculture, and was also a postdoctoral scholar in forest ecology at the same institution following her graduation. Her research and teaching have taken her across the Western US into Mexico, Costa Rica, Europe, and Indonesia. And when she's away from her office, Dr. Warren can be found exploring the outdoors, gardening, and reading. And with that, I'd love to turn it over to Kristen.
Thank you very much. I'm really excited to be here today. Let me share my screen. Does that look good?
It does.
Okay, great. Um, so thank you all for coming, and I'm really excited to talk about the species that I've been working on with hopefully some new information and also probably some information that you've heard before. So I work on Southwestern white pine, and what I'm going to cover today are some highlights and some management implication and recommendations for what we've worked on over about 15 years now. So it's pretty exciting to be at this stage of the research. Um, so before I get started on the research, I do have a few acknowledgements that are really important to me to cover. First off, I want to just acknowledge that where we are in Flagstaff, we're at the base of the San Francisco Peaks, and this area and across the Arizona and New Mexico and the Southwest are homelands sacred to the Native Americans, and that we live and work on their homelands. And so I think it's just important to recognize and acknowledge that. This is a really large international collaborative effort, these different projects that I'm going to cover. None of this would have happened without all of these different people. So I've been the lead PI on a lot of these projects, but there's a whole set of other principal investigators that work with me on these and lead different components of it. There's been a whole set of graduate students and postdocs and staff, as well as undergraduates, field and lab technicians, and we've had some citizen science efforts along the way. We also have a lot of collaborators who just facilitate the work or who have been involved in specific parts of different projects. So without all of these people, none of this work would have really happened. So it's really fun to be part of a really passionate group of people who work on these topics and who work together really well. So that's been really fun. And then finally, the funding is also key to getting all this work done. So we've had a suite of different funding from the USDA Forest Service, from internal NAU programs, and then also through the National Science Foundation. And that National Science Foundation funding was critical to a lot of the work that I'm going to talk about today. So, but it was built on the backs of a lot of Forest Service funding that has helped and been pretty continuous along the way. So. All right, so with those done, I want to introduce you to our tree. So we're going to talk today about a lot of different projects. And so you may end up with a lot of questions. I'm going to highlight a lot of different things that kind of some details, but some superficial stuff just because of time. So we started by looking at the silvics ecology, physiology, and silviculture of the species. Then we added genomics and some hybridization work in there. A lot of this is also ongoing. We've looked at disease resistance and the durability of that resistance in the landscape. We're looking at disease monitoring, detection, and spread, as well as climate change factors, including range shifts and adaptive traits, and tying all those together and looking at interactions among all these different research categories to look at how we might manage the species into the future, because that's really, as a silviculturist, what I'm really interested in the applied aspects of our research. So if you're not familiar with Southwestern white pine, it's Pinus strobiformis. It is a species that grows in the US Southwest. So this is Arizona, New Mexico here, and then down in this map, the southern part is Mexico. And a couple of things I want to highlight. So the red polygons on this map are the results of an ensemble model that we developed several years ago to predict where we would find Southwestern white pine in the landscape, because we found that the little range map, which are the gray polygons, didn't adequately describe where we were seeing Southwestern white pine. And the red polygons do a pretty good job of describing where we find it. And that was based on presence absence points across the range. And so you'll notice that a lot of the range is south of the US border. So we do have collaborators in Mexico, and a lot of the work I'm going to talk about today covers the entire range of the species. It's important to note that when I talk about the southwestern portions and the research that's just taken place in the Southwest, US states, that's just the very northern portion of the range. So we have to always kind of keep that in mind, that we're in that kind of transition zone. That's going to become more apparent as I go through. The other thing I want to note is if you're not familiar with the Southwest, in this area of the southern part of both Arizona and New Mexico, and in some places outside of, in kind of central New Mexico, we have what we call sky islands. And those are mountain ranges that rise up out of the desert. And there's a gradient of ecosystems on those sky islands, including the habitat where we find Southwestern white pine. And so these sky islands, then those populations can be separated by wide expanses of desert ecosystem. And so that raises some special concerns about managing for things like genetic diversity. Okay, so then moving back over to these pictures. So this is on the left, Southwestern white pine, a mature tree. It's got pretty large cones. It's got large seeds in those large cones, so they're pretty nutritious and important for wildlife. However, we don't have Clark's nutcracker throughout the range of Southwestern white. We kind of lose the range of Clark's nutcracker in the middle of Arizona and New Mexico. So it's just really nutcrackers only in the very northern part of the range. It is an important food source, though, as you move south for other species. The dispersal agents and some of the dispersal ecology is still a research need that we have to pursue at some point. It also is a really important component of the mixed conifer just in terms of providing biodiversity and maintaining a functioning mixed conifer ecosystem. And the mixed conifer ecosystem that it's part of is kind of it's kind of a moisture sensitive species, and so it doesn't actually inhabit a wide ecological amplitude in terms of elevation range. It doesn't grow super high. It's not as high of elevation white pine as, say, bristlecone pine in the Southwest. And it doesn't do all that well when you go lower in elevation into sort of the ponderosa pine zone. It's not super tolerant of fire, especially when it's young and those really dry conditions. So it's a bit of a specialist species. And then the other thing I want to note is this, there's a seedling picture in the middle here that it's shade mid-tolerant. So in terms of its ecological niche, it's fairly similar to Douglas fir. Everywhere we look for it in the Southwest, we find it adjacent to Douglas fir in the mix. The exception to that is this one little population over here in Texas that does not have Douglas fir. So it's a mid-tolerant species. And it does do better when you open up the canopy. It grows better, but it will survive just fine in fairly shady conditions. Okay, so that's a kind of an introduction to the species we're interested in learning more about today and what we've been focused on. I want to talk about the genomics next. And so if you have specific questions about a lot of these different factors, please ask them, and then I will redirect as need be. So I'm not a geneticist, I'm not a pathologist, I'm not a lot of things. And so again, I might redirect you to specific people if you have really complex questions about some of these things. One of our previous graduate students on this project, Mitra Menon, looked into the hybrid complex that exists between Pinus flexilis, which is lumber pine, and Pinus strobiformis. So these two species have been known to hybridize. This has been in the literature for quite some time. But what we found was that the hybrid nature is far more prevalent than I think we thought it was. You really don't find pure genotypes of either species when you're in Arizona and New Mexico. And so there is this genomic cline, and there's also a morphological cline, and they're not in the same geographic location. So you can see that on this map. And so we redefined this hybrid range with this pink polygon here. And if you look over to the left, the brown bars here indicate what the Pinus flexilis genes are. And then the purple bars are Pinus strobiformis genes. And anywhere you have a bar that's not either purple or brown, but has both, those are the hybrids. And the entire area of the US Southwest is the hybrid complex, except for when you're very far south near the Mexican border. And when you're up into southern Colorado, you start to get into pure Pinus flexilis genes. Morphologically, they're incredibly difficult to tell apart. We've tried to do that in the field. It's just really challenging. So this has, in and of itself, it has management implications, but we've also found some really interesting things related to this hybrid complex. Mitra looked into gene flow over an evolutionary time period and found that there is gene flow happening between the hybrid zone and pure Pinus flexilis further north. And I didn't put the strength of that flow on these arrows, but the gene flow is stronger going from strobiformis into flexilis than the other way around, which is pretty interesting. And so that hybrid zone is kind of moving north, which corresponds with that genomic Klein center. We did look for connections with some environmental variables and found connections with freeze traits that come from flexilis, drought traits, and adaptation to hotter environments that come from the strobiformis, and then also with growth traits. And that this, especially this last piece, this ongoing work as we look at dendrophenotypes. So we use tree cores to look at these growth traits for the parent trees for a lot of the seedlings that we've been studying. And then in this hybrid zone, there's a climate gradient that corresponds with the hybrid matrix as well. So as you move from south to north, you steadily increase the amount of flexilis genes you find in the population. And that corresponds with a temperature gradient. So it's warmer in the southern part of the hybrid zone, and then it gets colder as you move north. There's also a monsoonal pattern. So we have summer monsoon rains. That rain signal in the summer is strongest near the Mexican border. And then as you move north, you get into more of a Rocky Mountains kind of precipitation pattern, and you lose that monsoon signal. It's less strong. So there's a lot of correlations between the genetics of these two species and this hybrid zone with these different environmental traits, which is kind of exciting from a climate change perspective. One of the hypotheses that we put out in these papers is that this hybrid complexes and the linking of these different traits is potentially going to make this hybrid zone really key for climate change, that these hybrids might actually fare better under climate change scenarios in the future. You know, we'll have to wait and see, but it's pretty interesting stuff. Okay, so I kind of touched on the climate change threat already, but we view this as kind of three big threats to the species, and they're probably similar to the other white pine species if you've been working on those and looking at the threats to those species, but climate change, high severity fires, and then white pine blister rust. And I'm going to talk about research in all three of these categories, but also some research that spans one or more than one of these categories as we move through. Looking at climate change a little bit more in depth first. So the Southwest and Mexico are projected to get warmer and drier. They already have gotten warmer and drier. They're going to get more warmer and drier. There's also going to be hotter droughts, which puts more stress on the trees. We've already documented pretty widespread mortality events in both Mexico and the Southwest from these drought events. Those are going to continue to happen probably with increasing frequency. So with the same set of data points we use to develop the ensemble model for where the species exists now, we use those data points to do a climate envelope model, which just looks at where we find it now, and then under different climate change scenarios, where we might find it in the future. And this map here shows where we project contraction to happen. So the orange and red, depending on the different climate models that we used, more or less contraction. And then also some areas where we predict maybe more stable. So we don't think that white pine will completely disappear from Mexico, for example. There will probably be some areas of refugia. In the northern part of the range up here, we expect it to have some expansion potential in terms of the climate that's suitable for the species. And again, that area of expansion will depend on what actually happens with the climate. What's really interesting here is that hybrid zone in the middle, where we think there will be both opportunities for expansion, but also there will be contraction of the range. And given what's going on genetically with the species, it's going to be fascinating to see how that plays out into the future in terms of which genes and gene complexes do well in future environments. Okay, so moving into fire then, I want to talk about high severity fire a little bit, and I'll give you some results from our work in this related to white pine. So in the Southwest, like other parts of the West, we have had increasing fuels and tree densities since around 1900, largely related to our management practices in the forests, both from a silviculture standpoint, but also fire management and really removing or suppressing the good fires in the landscape. And that's true of our mixed conifer as well. There's two different types of mixed conifer in the Southwest. There's dry and they're wet. I don't have time to get into the details between those, but there was more frequent low intensity fire in the dry than in the wet, but both had kind of a mixed severity kind of fire regime historically. This has led to a species compositional shift towards more fire intolerant species. Down in the US Southwest, that means increases in things like white fir and Douglas fir, and also actually increases in the white pine because it is fairly fire intolerant. So we start to see it growing underneath ponderosa pine, for example, where frequent fire would take it out, and we wouldn't see it there very much. Stand replacing fires have increased in size and frequency. There is some interaction with climate change here. Really, the point is, though, that we have more high severity fire in the landscape, and that has a big impact on those, especially those small populations we find on the mountaintops and the sky islands. So we have the potential to lose an entire population in one high severity fire event. We haven't seen that yet, but there's other implications for having high severity fire as well. So this is really seen as kind of a problem. Okay, so we started installing permanent monitoring plots across the landscape back in 2010. And the intent of the original network was to track white pine blister rust in southwestern white pine. However, shortly after we started installing the plots, they started burning down. And so over the subsequent decade, 27 plots burned. 15 of those burned completely in high severity events. 12 were low to moderate severity events. That's as rated by composite burn index, which we did a quantitative assessment of in the 1-year post-fire assessment. So for all of these 27 plots, we have 1, 5, and for some of them, we have 10-year post-fire data, but not all of them because it hasn't been 10 years yet. So I'm going to talk about these red points on the map. Those are the ones that burned. I'll come back and talk about the green points in a few minutes. Okay, so on our low to moderate severity plots, this is what they look like. So 2012 was 1 year post-fire. So we had the understory was impacted the most. And by 2021, that understory had fallen down. And so the conditions were pretty open. So this is a relatively positive shift in conditions. I like the structure that we have after 10 years post-fire, way better than the structure that existed there pre-fire because of the fuel conditions. So the overstory mortality was pretty limited. It did continue. So we did find the 10-year remeasurement was critical to capture all of the mortality that was related to the original fire. We also found conifers across all of the canopy layers, which is an important finding relative to what we found in high severity. This includes the white pine. So everything was regenerating pretty well in these in this post-fire environment. There was some buildup after the fire in 1,000 hours and smaller fuels, but not a significant proportion and not enough to be really concerned about. This one we're concerned about a little bit, increases in ribes shrubs. So ribes are the alternate host for white pine blister rust. And that means if you increase ribes, you may increase incidence of white pine blister rust. So I'm going to pull in a figure here. So I'm going to explain this. So this is on the left side of this figure is high severity plots, and on the right side are the low and moderate severity plots. And where there is the red asterisk, those are where we find a significant differences across time. So on the x-axis is time and our different remeasurement periods, and then the number of ribes stems per hectare is what the bars are showing. And in the low to moderate severity plots, we didn't really have much going on in the way of ribes densities before the fire, and that was true in the high severity plots as well. By year 5, though, they had really increased. They've started to come down in year 10, so we're thinking that trajectory will probably continue, but we're probably still going to see a higher abundance post-fire of the ribes than we had pre-fire. So we're continuing to track these. On the low to moderate severity plots, this is more concerning than the high severity plots, where we saw lower abundances but the same trends in terms of build up to year 5 and then some lack of establishment essentially of those new shrubs. Because there's still white pine in the low to moderate severity plots, and that's not true in the high severity plots. So we'll continue to watch this through time as we do the next stage of monitoring and see if we start to see any changes in the amount of rust on these plots. And then I want to just for a minute talk about high severity. It's not as interesting from a white pine standpoint, but these were conifer dominated. They were selected because they had a certain amount of basal area of white pine in the overstory, but they often did have some aspen and some other sprouting species in them. So what's happened is we've seen the regeneration 10 years later dominated by non-conifer species, if we have regeneration at all. And so the overstory mortality was very high, approaching 100% on most of these plots, if not 100%. No regeneration of conifers, with the rare exception of some Douglas fir and ponderosa pine. And those plots were all close to an existing seed wall where the trees survived the fire. So there was the ability for those trees to reseed in on these plots. We saw a huge increase in fuel load that leads to a reburn potential that's very concerning. And the structure and composition of these plots was fundamentally altered, not always in a bad way. This plot here that has aspen dominated area. We love aspen in our landscape. It's also a species at risk under specifically climate change and a couple of other things. So, and aspen has always kind of ebbed and flowed in our landscape because of fire. So we welcome more aspen in the landscape. Although the size of these burn patches is pretty unusual. Other areas and plots came back dominated by Gamble oak and New Mexico locust, which are shorter statured. They tend to be shrublands instead of forests. And so we're paying a lot of attention to that in the Southwest, that we're having these ecological shifts into a different forest type. Okay, so moving into white pine blister rust then. So in the Southwest, this is Arizona and New Mexico. White pine blister rust is still moving across the landscape. So this, especially 15 years ago when we started this, there was this huge opportunity to look at areas that had rust and didn't have rust. And we continue to do that, but it has spread slowly in the last 15 years. So there's more places that have rust now than there were before. Over here in South Central New Mexico, those were the first identifications of rust in the Southwest. They have the identification year of 1990 and more recently, but if you actually date those cankers and look at their research, they date back to probably the 1970s. So rust has been in South Central New Mexico for quite a while. In Eastern Arizona, I'm going to call attention to these two areas because I'm going to show some results specific to these two areas. We didn't identify it until 2009. It had probably been there for about a decade before it was identified. And so this is a hot spot for intensification right now and spread. And so the rust situation, we have 10 years of those monitoring plots. So I'll just remind you of our plot network. We had originally 81 plots and 27 of those burned. So I'm going to talk about the green points are the remaining unburned plots after 10 years. And so that's what's coming next. If you're unfamiliar with the effects of rust on host trees, they form these cankers with these swollen growths on the branches and on the bole of the trees. The stem cankers are almost always lethal. That includes Southwestern white pine. It's pretty similar to how it reacts with as the other white pine species. And then rust can also cause foliar dieback, top kill, and loss of regenerative capacity. Okay, so what did we find? In our monitoring network, we did see about a 20% increase in the number of infected plots between the installment and the 10-year remeasurement. And that was largely happening in this Eastern Arizona area that has had more recent infection. What we found geographically in the South Central New Mexico area, the Sacramento Mountains, was that We didn't find much new infection on plots that didn't have infection before, but we did find intensification of the rust on the plots that were already infected. So in these pie charts, these represent a plot. This is the proportion of trees that have infections. The pink shows rust infections. The purple, the proportions of trees that we could tell for sure died from the rust. And then there's all this gray proportion where the trees died, but we couldn't say for sure it was from rust. There was a 10-year gap from when we first went out to the plots to when we came back and remeasured. So if a tree died not long after we installed the plot, there was no way to really tell why that tree died. 10 years later, however, if you look at this point up here on the Santa Fe and these points over here near Flagstaff where there's no rust, you also see that there's very little background level of mortality. So likely a lot of these gray areas where we have rust are due to that mortality is due to rust, but we can't confirm that for sure. Okay, in terms of individual host tree effects, this is just looking at the plots that have rust. This is first looking at just where we have branch cankers. So 17% of those trees had top kill, 15% crown dieback, 34% fewer new cones than uninfected trees, and the mortality rate of these trees is 1.6% per year. We add the bull cankers, it gets much worse. So about 80% of those trees had top kill. The mortality rate almost doubled to 2.9% per year. 30% mean crown dieback, so double the amount of crown dieback, and 55% fewer new cones than uninfected. So the impact of rust on the potential regeneration of white pine is pretty severe, even though rust is limited to just select areas of the Southwest currently. Okay, so just to bring some points home, we did find some new geographic spread and increases in incidence across the infected site. We think the geographic differences we found are due to the time since infection in South Central New Mexico versus Eastern Arizona, those two hotspots. Those are the two different epidemics. I didn't show this result because I don't have enough time, but the climatic and site factors, we did find some of these were important in predicting incidence. And this work for both the fire, the post-fire, and the white pine blister rust plot remeasurements was part of Simon Baker's thesis. He just finished last May. He's working on getting both of his publications or his manuscripts ready for publication. So this will be forthcoming in the literature, hopefully in the next year or so. And then just because it's white pine blister rust, it takes a while to kill individual trees, and Southwestern white pine is often not a major component of the overstory or the understory in a lot of these stands. The stand structural changes are really pretty minor right now. I expect that those will increase in the future as we see more mortality from rust, but it's still going to be subtle because of the nature of these ecosystems and the nature of rust and how it slowly kills trees. And then finally, we are concerned about the higher levels of mortality and the potential loss of regeneration in those areas that have rust. Okay, so moving into genetic resistance testing, I'm ping-ponging to give you a kind of a broad overview of what we've been working on. So the genetic resistance testing happens at the Dorena Genetic Resource Center up in Oregon. And the leads for this piece of the project are Richard Snesko and Jeremy Johnson. Jeremy started as a postdoc on this project. He's now faculty at Michigan State University. And the results I'm going to show today are a work in progress, so they haven't been published yet, but we're working on getting these ready for publication as well. So we've been testing for both major gene resistance, which, if you're unfamiliar, is a gene for gene relationship. So it essentially confers immunity to seedlings that have that gene or trees that have the gene. And then quantitative disease resistance as well, which is polygenic and is considered more durable because the pathogen can't mutate and overtake that gene so quickly. But the trees will get symptoms and survival rates are lower than you would have with the major gene resistance. So we're looking for both. And what they do at Dorena is they, if you look at this left-hand picture, they grow the seedlings into and put them in these raised bed boxes. And then in the fall, they go out and collect ribes that are infected with the rust. And over in this picture, you can see a picture of the spores on the underside of the ribes leaves. So they put the leaves face down, so the spores are facing the seedlings on these mesh racks in their inoculation chamber. They moderate the temperature to be relatively cool. So there's a specific temperature requirements for rust inoculation of the seedlings. And then they bring the humidity up to 100% and essentially rain spores down on the seedlings and inoculate them that way. And you can see that there's some plastic bags here covering seedlings. That was so that we could have a few seedlings from every family that weren't inoculated in these direct inoculation trials. And then the seedlings are moved back outside into the raised bed gardens and tracked by the staff and in a very time and labor intensive process to count needle spots and different bark reactions and different cankering, and given a rust severity score over several years and different inspections. So we were able to include the entire range of Southwestern white pine in the testing. There had been some limited testing prior to this, but this was a really big effort to increase the available rust resistance information that we had for this species. So in total, over several years, we've tested 430 families just in these 3 sow years that I'm showing here. So 2014, 2016, and 2017. And there's kind of a geographic split here in the map where I've got the US population circled and then the Mexico population circled. One thing that I forgot to note when I showed the map of rust was that there is no rust in Mexico yet. And so when we're testing for disease resistance from the Mexican populations, we're collecting from trees that have never seen rust and populations that haven't seen rust. And so they're naive. There's been no selection pressure from the rust on the population. So that's just a really important thing to note when I show you the results, because it comes up again. So of these different families, we had thousands of seedlings in these trials. The MGR trials, that refers to the major gene for resistance trials. And then the other 3 at the bottom with more seedlings involved are the quantitative disease resistance trials. So I'm just going to show you a couple of results that I guess they're not full results is what I'll say. Looking at quantitative disease resistance first, and I know these figures are pretty busy, so I'll walk you through it. So on the top set of graphs, these 3 on the top, that's percent survival of the seedlings. Each line represents a family. And then the blue graph is sow year 14, golden is 16, and then the pink and purple are sow year 17. And that sow year 17 includes the Mexico seed lots. And so in the percent survival, we're looking at higher levels of survival. So if you have 0 survival after you're inoculated, that means you're going to be really susceptible to rust. You probably don't have any quantitative disease resistance. And then if we look at average severity down here, this is the scoring of the rust rating at Durina. And so we want lower levels of severity when we're looking for quantitative disease resistance in the population. And so the bottom line of all 3 of these sow years and these results is that there is some families in every sow year that have both a higher level of survival and a lower average severity rating. So we are identifying trees that have quantitative disease resistance, which is pretty exciting. And then of this, in the 2017 sow year, that was 124 different families from Mexico, and 20 of those families had greater than 20% survival. And that's pretty exciting in a huge amount of population that hasn't seen rust. So that's a pretty cool result. So to dive into that a little bit more and show you what we would expect to see from major gene resistance as well. This is just percent survival and just a few selected populations from Mexico and 2 MGR populations from the US. So these are 2 different families that have MGR. This is quantitative disease resistance in the Mexico families. So that's, again, pretty exciting to find that. You can see this one susceptible goes down to 0% survival by the 4th inspection. I want to highlight this family here that's gray. It's RSCHI Ritchie 1151 that is a potential candidate for MGR in Mexico, which is also very exciting. And then to look at the average severity, which is the other metric I showed on the previous graph. Again, you want lower levels of severity instead of higher levels. And so that those fully susceptible family, you're at high levels of severity rating. But again, some potential QDR here. And then here's that Ritchie family here. That's a potential MGR from Mexico. So the fact that we were able to find some resistance in the Mexico populations has been pretty exciting for all of us and will help us to better manage, provide recommendations to the Mexicans when rust finally makes it across the border. And I strongly believe that it's a when and not an if. It's going to get there. It's just a matter of time. So, all right. And then the final note about the resistance testing comes from this remote sensing work. So Maria Hagsma was a graduate student at Oregon State University, and she worked with Jeremy and Richard to develop this hyperspectral rail system to look at whether we could use remote sensing to detect a disease incidence and how sickly the trees were. Remember, we had those plastic baggies over some of the seedlings to protect them from getting inoculated, and she found that we were able to detect the disease remotely. So she took the hyperspectral imaging that was done with this automatic rail system and camera and used machine learning to help detect and get some thresholds related to whether the trees were diseased or not. From a practical standpoint, this isn't a very portable system. It's not very useful in like the field, but a place like Dorina could potentially use it to help reduce the time and labor involved in the scoring when we're doing the rust resistance. So that could help speed things up in the future. Okay, common garden, I'm gonna have to speed up because I'm gonna get behind. So we had this common garden study at NAU, and we built upon this, what's called the Southwest Experimental Garden Array, to evaluate genetic variation, specifically under climate change. So we used an elevational gradient to simulate warming at 3 different elevations. We used a high elevation, a mid elevation, and a low elevation to do this. And we used a subset of the families I just talked about that were tested for disease resistance. We grew those in our common gardens. So we have a lot of information about these about these families. Okay, the experimental design in this set of gardens, this is the overhead view. We had a lot of raised bed garden boxes at each site. We had a weather station at each site and an irrigation system set up to water the seedlings as well. We had a lot of different replicants for each family. We had a lot of seedlings for several years out there, and it was a completely randomized design. So this was a really cool experiment that we were able to do. We also had a drought experiment superimposed on the top of this, where we had rainout shelters on half of the boxes, and we reduced the water to half of the ambient water coming in, and reduced the irrigation water as well. And that was just during the growing season. So we were out there generally we would put the irrigation and the rainout shelters up in about May and take them down in October. So this is just a snapshot of what these look like. So this is the high elevation garden, and you can see some annual temperatures and frost-free period. Here's the middle elevation garden, and then lastly, the warm and dry garden in the pinyon juniper ecosystem. And all of these temperatures match where you would find Southwestern white pine on an average annual basis, but that led to some understanding about the nuances of climate that were affecting our seedlings. I'll talk about that in a minute. So I just want to highlight some of the seedling performance things. So we were able to look at survival for all of these seedlings. We looked at height and diameter growth. We have some physiology measurements for a subset of the seedlings. We have bud burst and bud set phenology measurements for all of them. We also have carbon isotope data that we haven't yet analyzed for all the seedlings for pre and post drought. So we have some really good information on climate adaptive traits from these gardens. So we did get high survival across all the gardens and the drought treatment, except for the 2017 planting. Come back to that in the next slide. We did find that the seedlings had higher water use efficiency under the warmer, drier conditions. This was pretty exciting finding because it means that it's a plastic response in the species, so that under climate change, they might be able to respond and use water more efficiently. And we did also confirm that this species avoids stress through stromatal closure. And we found and confirmed faster growth, but higher mortality in seedlings from more southern populations. And that was first identified in a greenhouse study that Betsy Goodrich did several years ago, and then confirmed in this Carmen Garden study. And then finally, with the phenology data, we did find that early bud set in the seedlings led to increased survival. And that was particularly important when I talk about, on the next slide, the 2017 planting. So 2017 was the year that we planted the Mexican seed lots. And what we found was that if we planted seed sources from below 30 degrees latitude, they had a 15% survival in our gardens. And our garden locations, I didn't show the map, but they're located even further north in Arizona than Flagstaff. So they're up kind of near the Utah border. Above the 30-degree latitude was 80% survival. And we did have, so that 30 degrees latitude corresponds with the Mexican border. So we had 3 Mexican populations that exhibited about 50% survival across all 3 of the gardens. And so these are the 3 populations that we continue to play with in Arizona and New Mexico. The other ones we'll sometimes use, but we know that they're probably going to have low survival this far north. So we essentially moved these seedlings too far north, and we think it was early fall frost that got them. So that fluctuation in the shoulder season and that relationship between survival and bud set is probably what got them, not the overall temperature. Okay, so we also have field durability trials going on. So the intent of these initially was to examine stability and durability of the rust resistance. So, you know, how stable is it going to be in our landscape? We also want to monitor for potential virulence to the major gene, which we know is out there, is just identified in limber pine recently up in Canada. So we want to make sure we're monitoring for that. We've also been using it to monitor the survival and validate the adaptive traits that we identified in the common gardens. So that's been pretty exciting. We've been able to continue planting all these seed lots out and identifying the most promising ones that might have a suite of good traits that we want to capitalize on. And then this past year has been pretty exciting for me as well. We added research seedlings to operational plantings, working with the managers in 2024. So the first set of plantings were in these fenced common garden kind of style plantings. And now we're actually incorporating them into these operational plantings, which just means that regardless of what our outcomes are from a research standpoint, some of these seedlings should have some resistance to rust in them. So it's good to just get those out into the landscape. Okay, so I'm going to wrap up by talking about some additional needs that we need to continue working on in terms of research, development, and monitoring, and then talking about management implications and recommendations. So we don't have regional hazard models yet for white pine blister rust, and we need to develop those. We think we probably have sufficient data to do that now with our 10-year rust remeasurements and the available climate data that we have. We just need to find some additional funding to move that piece forward. We also need some enhanced early detection and monitoring efforts, and this may come in the form of remote sensing work. So we need to continue pushing that envelope as well and using technology to do that. All of the research that I've talked about, we've identified some holes and some things we still need to know about the ecology of the species, physiology, the effect of climate, and continue to identify traits that will help this species, will help us manage it into the future, essentially. We need more disease resistance screening. So there was another year in 2020. We're trying to continue this process. There are local managers who want to operationally put resistance out on the landscape, but they're unable to because we either haven't identified resistant trees on their district or haven't collected seed and included it in trials yet. So this is an effort that needs to continue. I didn't talk about it, but we have done some genetic conservation for the species and put seed into long-term storage. But that is an effort that also needs to continue, especially as we learn more and more about the genetics of the species. In terms of general strategies for managing for resilient stand structures that include white pine, more open conditions are going to be good from a variety of perspectives, but from a white pine perspective in particular, lower basal area we know helps increase the growth of understory white pine. In particular, we want to get them up into the overstory so they start producing seed. It will also help with abundance of regeneration, and we have data to show both of these things. I'll talk more about specific strategies for regeneration on the next slide, but in general, we just need to plan for white pine regeneration. It shouldn't be something that's just assumed will happen. Fire hazard reduction goes right along with managing for open conditions. You can see in this image on the left that there's pretty high fire hazard there. So lots of ladder fuels, lots of fuel buildup on the forest floor. Even overstory white pine, unless they're pretty old with pretty thick bark, will succumb to fire events, even if it's fairly low severity. So we see that in our data. White pine continues to die at a faster rate than both Douglas fir and certainly ponderosa pine. So it's a little less fire tolerant than other species. So we need to protect these trees by reducing the fire hazard around them. And this one is something that I'm pretty passionate about, saying that if we don't account for white pine blister rust cause mortality in management, we're not going to be able to sustain the species. And what I mean by this is that historically, the species was a pretty low percentage of the mixed conifer forests, and that over time, it's actually increased in abundance because of removal of low intensity surface fire or fire at all, and the way that we've managed these forests. If we try to, so in the Southwest, we manage a lot for restoration objectives, which is great, except those restoration objectives say go back to historic species compositions. If we do that and we remove most of the white pine from the understory, we are not accounting for the impact of rust on those stands. And so we're not going to have enough regeneration. We have to allow for the rust mortality to happen and still have enough understory trees to make it into the overstory. And we have published specific numbers for different parts of the Southwest in this Goodrich paper from 2018. So this is something that I think is really important and easy for a lot of managers to ignore because rust isn't in their local area yet. So it's easy to not think about it. Okay, so specific regeneration recommendations. We need to think about our seed movement. I made some points about how we killed all of our seedlings in 2017 that we moved up from Mexico. Those big geographic jumps we don't want. We also know that Southwestern white doesn't do very well on the driest sites, so there's no reason to really plant them on dry sites. We also have some thresholds for how far we actually can move them. This paper hasn't been published yet, but it's from a dissertation from a few years ago. So we still have some work to do on publications. We're going to combine this climate adaptive traits information with genetic resistance information. We're all still working on this piece because the resistance trials have just ended fairly recently. And then in terms of prioritizing planting, that we should have the rust resistance and drought tolerant stock available to plant in the post-fire areas. There's a lot of post-fire planting happening in the Southwest. And we also want to plant in areas with high white pine blister rust incidence. We can focus more on natural regeneration where we have healthy stands still and areas where we have verified disease resistance because those trees are producing seed and where climate may be less problematic. So if we have areas of refugia. In general, all management should retain Southwestern white pine without lethal cankers. So that's to maintain genetic diversity and also any trees that might have QDR, the quantitative disease resistance. And that is a shift in management that I've seen happen over the last 20 years. When I first started working on this species, managers tended to want to remove any tree that had any canker, any rust at all. We want to prioritize protection of our plus trees, so those trees that have genetic resistance or have really good drought traits, other adaptive traits. We want to protect those from fire and also from harvest. This is something we've been communicating with the Forest Service. We have lost some of our trees, our sample trees, to both fire and harvest over the years. Potentially need to consider those increases in rabies I mentioned with the fire. Just pay attention to that. And then we should always be thinking about all three of these threats: white pine blister rust, fire, and climate when we're managing the species. The landscape perspective is also really important. What was our historic variability in the landscape? What's the future variability? Where do we have these different traits across the landscape? Where might we have fire refugia or climate refugia? There's a lot that goes into thinking about appropriate management at these landscape scales. However, I think that largely we are pretty positive thinking about the species. We found some really good and interesting results. We found the resistance. We found some climate adaptive traits that are pretty interesting. So we feel that we really can be proactive and manage the species to protect the ecosystem services provided by these really important forest types and the species in particular in the Southwest. So I think that it's a nuanced story and one that I've been really happy to share with you today. So hopefully I didn't move too fast, but I know I covered a lot. So thank you.
Thank you so much for your talk, Kristen. Now I'd like to open the opportunity for our attendees to share your doubts, comments, and questions. Remember that we only have about 4 minutes left. That's good enough for 2 or 3 questions. Please type your question in the comments on this chat section. And if we don't have enough time to go over all the questions, don't worry. Our team will send them to Kristen with your contact information for further discussion. And let's see. Okay, so we have the first question from Ras Oaks. It says, how much rabies is in there in Mexico?
That's a good question. I haven't really looked for data on rabies in Mexico. Generally, there's not as much information available about the forest composition in Mexico. You can find tree compositional information fairly easily, but not so much for the rabies. I know it's there. I just don't know about the abundances or even how many species they have. So it's a good question, though. So now I'm going to have to go ask my colleague if he has a good answer for that.
Nice. The next one is from Kevin Potter. He says, excellent presentation, Kristen. Can you talk about the need and challenges of working across an international border to understand the genetics of these species and to manage it into the future? It seemed that Mexican seed sources, for example, could be helpful for maintaining and restoring the species in the Southwest.
Absolutely. That's an excellent point. So we had to work through a lot of permitting early on. It took us a full year after they had collected the seed to get the seed across the border. And so that was a challenge, but we figured out how to work through it. One of the issues was that the species was being mislabeled by the Mexican authorities as a different species that fell under CITES, and strobiformis doesn't fall under CITES in Mexico. It's not threatened at all in Mexico. It's quite abundant. So that was an interesting thing that we didn't anticipate happening. Working directly with Mexican collaborators is absolutely key. And I also fully agree that we absolutely need to be working across these borders. They're political boundaries. They're not ecological in any way. And so if we don't do that, we're doing our ecosystems a disservice. And I would argue that's the same for the Canadian border, that we all need to be working across these boundaries in order to manage appropriate under climate change, these species that span these international borders.
Nice. And the last question for today, and just to remind, we're going to send all the non-addressed questions later to Kristen, but from Cindy Smith, are there any insects threatening this species?
There's no insects threatening it. It does get attacked by mountain pine beetle, but I haven't observed widespread mortality from mountain pine beetle. There's also a lot of seed and cone insect activity, and in some areas during certain years, you can lose all of your seeds essentially to seed and cone insects, which can be kind of devastating if you're trying to collect in that area during a certain year, which just begs that we should just have seed collections in storage for our reforestation needs and for things like genetic testing. Just build up those seed banks so that we're not dependent on a specific year collection.
That's interesting. Well, with that, we're going to close the Q&A session. And on behalf of the WebArc Pine Ecosystem Foundation and the webinar organization team, I want to thank Kristen again for your participation. And our next webinar will be on January 21st with Lacey Hankin presenting her work on soil moisture mediates 5-needle pine seedling mortality under extreme heat conditions. Thanks again, everyone, for attending, and stay tuned for our upcoming installments. And happy holidays, and see you next month.
Thank you very much.
Bye-bye.
Bye.